Hello, my name is Caitlin Dickinson, and I am pursuing my graduate degree in biomedical engineering, and my thesis is investigating the cellular uptake of PEG-related nanoparticles conjugated with cell-penetrating peptide. So as we know, cancer is currently the second leading cause of death within the United States, and according to the National Cancer Institute, lung and bronchitis cancer account for approximately 12.7% of all new cancer cases and 22.4% of all cancer deaths. Our current method of treating lung cancer is through the delivery of chemotherapeutics. However, this method can be particularly difficult on patients due to it being nonspecific, and that means that healthy tissues within the person can also be negatively impacted by the drugs that you use, as well as the tumor. Due to this, drug delivery has been of high interest, and there have been a lot of investigations to explore how we can enhance the current systems that we know. One of the most common techniques is the use of pegylation. This is the addition of polyethylene glycol to a nanoparticle or drug surface. And not only does this help stabilize the cargo within the human body, the increase in size also helps to prolong the circulation and having more resistance against renal filtrations, and as well as reduces the nonspecific particle uptake. There's also been a new growing field using cell-penetrating peptides. These are defined as short amino acid sequences that are between 4 to 40 amino acids in length. Actinipedia has been one of the most researched cell-penetrating peptides since it was found from the homodomain of Drosophila in 1918. The peptide was found to be the minimal sequence needed that can facilitate cellular uptake, and has been increasingly explored because it has a very low toxicity despite when used at high concentration, and has a large cargo carrying ability. While both these have been investigated heavily in their respective areas, neither have been fully investigated together, which is where my research begins to fit in which I'll be exploring and optimizing the ratio of CPP to PEG. The first step will be to assess the surface characterization of my nanoparticles. First, we'll start with a carboxylated fluorescence polystyrene, and then we'll have that in addition of PEG, polystyrene in addition to cell-penetrating peptide, and then polystyrene in addition to both the PEG and cell-penetrating peptide. These will then be characterized through dynamic light scattering as well as scanning electron microscopy to assess the size, zeta potential, and morphology, since these are all factors that can alter the cellular uptake. Once this is done, the nanoparticles will be added to a 24-well plate in triplicate with either A549 cells, which is a lung cancer cell line, or a THP1M1 cell line, which was very phagocytic, which will be used as our positive control. After that, the cellular uptake will be characterized and analyzed through fluorescence, confocal, and multiphoton microscopy. The three techniques will enable for a more holistic assessment of the nanoparticle uptake, and we should be able to test the size limitations. So special acknowledgement to my advisor, Dr. Brenza, and the laboratory group, the biomedical engineering program, and the chemistry, biology, and health sciences department. Thank you.